This is the first short video related to blood, and this is really meant to be an overview and this is for human anatomy and physiology too. So the first thing that I want to do is I want to give you some references. If you like to follow along in the Saladin textbook, this is the 8th edition, then you'll want to look for section 18.1, and specifically you can look at pages 671 to 673. If you're looking in the WISE lab manual, I'm afraid WISE lets us down a little bit. I can refer you to exercise 24, but he really doesn't have a very good overview. So what I've done instead is I've given you a link here to OpenStax, which does have kind of an overview, which is pretty handy. And I've posted this on ELC as well. And I've also got a link here to Wikipedia, and that's on ELC also. Just a heads up, the Wikipedia link is extensive. So there are discussions of blood cells in animals that are not mammals. There is discussion of old-time bloodletting techniques. There might even be a reference to a vampire there. So the Wikipedia entry will really be way more than you really wanted to know about blood. All right, so let's move on a little bit. Now, looking at the need to knows, what I do want you to see is that the purpose of this video is actually to introduce you mostly to some vocabulary. Most of these terms here relate to the blood or the formation of the blood. We have a couple that are related to some different conditions where we've got blood dysfunction or some disruption in blood formation. And then we have a couple of other terms here that relate to something about the blood. So our purpose really is just to give you an overview. Now this image, which I actually took from the OpenStax textbook, shows you in kind of a cartoony way a tube of blood. Maybe this would be a tube of blood, say, that would be drawn during a regular doctor's visit for some diagnostic purpose. And in this particular tube of blood, what they've done is they've centrifuged it. So the heaviest elements will go to the bottom. And if you look here, notice that the heaviest elements at the bottom of the tube are going to be our red blood cells, and then something called the buffy coat here. And the buffy coat is going to be composed of white blood cells and platelets. Now the lightest element then is going to be the plasma. This is going to be this straw-colored fluid that you see here at the top of the tube. The plasma comprises about 55% of the total blood volume, and about 90% of that is actually water. So the vast majority of plasma is water. There are a good number of proteins that are present there as well, and other chemicals and electrolytes, other hormones and chemical messengers. So we said here that the cellular elements are going to be here at the bottom, right? Our red blood cells and our white blood cells here are all going to be at the bottom. We have a particular name for the cellular components of the blood. We call them the formed elements. So a formed element is going to be anything that is a cell or is derived from a cell, and that would include erythrocytes, which are red blood cells, leukocytes, which are white blood cells, and platelets, which are cell fragments. Okay. The formed elements form about 45% of the total blood volume. All right. Now the reason that the red blood cells are the heaviest thing here, you may remember, is that the red blood cells contain hemoglobin, and hemoglobin contains iron. The hemoglobin and the iron is actually the protein. Those are the elements within the red blood cells that are responsible for binding to oxygen so that the blood can carry it to tissues where it's needed. We have a particular term that we use to describe the volume of the blood that's composed of red blood cells. We call that the hematocrit. And that really is just a sort of a technical term that refers to the percentage of the volume that is red blood cells in this case. All right, so let's look now. Let's move to a slightly different figure here. In this particular picture, what we're looking at are two examples of conditions that can result from some disruption in the formation of blood. In this tube here in the middle, we have anemia. This would be one example of anemia. There are actually several types of anemia that people can develop. And anemia, the name actually refers to, what it means is without blood, anemia. And if you look at this, you can see that even though the tube contains the same volume of blood, the percentage of blood that actually is composed of red blood cells is going to be much smaller. In a contrasting condition, polycythemia here, we have a condition where in the same volume of blood, we actually have a relatively greater percentage of red blood cells. And this is what the word polycythemia means: many blood cells, many blood cells. Polycythemia and anemia can have many causes. So there are several diseases that can be associated with either anemia or with polycythemia. But you can see then that the ultimate change is actually in the hematocrit, the percentage of red blood cells that are present in each tube. All right. This is a very cartoony picture of the formed elements of the blood. And so what we're looking at here, just an artist depiction of the different cells that are going to be present, those cellular elements. So we have red blood cells, and you can see here they've got a very distinctive shape. We call it a biconcave disc, so they look a little bit like cough drops. Red blood cells also have no nucleus. They're essentially cells that are filled with hemoglobin molecules. And the hemoglobin, as I said on the previous slide, is that protein that's responsible for binding to oxygen and delivering it to tissues that are working. Platelets are going to be our second formed element here. These are cell fragments. They break off from much bigger cells that are called megakaryocytes. Platelets have important functions in tissue repair and in blood clotting. And then down here, we see that we have our white blood cells, and you can see we've got several types of white blood cells. And this artist is showing us a monocyte, showing us a couple of different lymphocytes, an eosinophil, a basophil, and a neutrophil. And I do want to just point out that these have been sort of, colored for illustrative purposes. We can divide our 5 types of white blood cells into 2 big, broad categories. The first category would be A granulocytes, and that would include monocytes and lymphocytes. And the other big category would be granulocytes, and that would be our eosinophil, our basophils, and our neutrophils. The granulocytes, as you can see here in this drawing, will have granules that are prominently visible in the cytoplasm, whereas the A granulocytes lack these granules. That is the big defining feature between these 2 big categories. All right. Now, what I've done here is I've actually taken and rearranged a table that shows us the formed elements. Here we've got the relative prevalence of each of these, some description of what they appear, a function, and then a little comment here. I've posted this on ELC for you, but I just wanted to make a couple of comments about it. So with the erythrocytes, you can see that they're going to be the most prevalent. Here's a description of their shape and their job. As we said, red blood cells are going to have the job of transporting oxygen. Platelets are also going to be relatively prevalent, and their most important function is going to be in hemostasis, which is a fancy word for blood clotting. Starting with our granulocytes here, you can see we have neutrophils, eosinophils, and basophils, and the relative prevalence here, an appearance, a description of the appearance here, and a summary of their functions. We're going to be talking more about these in the next video, so stay tuned for a second video. And then here we've got the A granulocytes, your lymphocytes, and your monocytes. And again, this is so handy because you can see the relative prevalence as well. All right, this is a blood smear, and I made this image from this really great resource that we have from the University of Michigan School of Medicine. And in this particular site, you can actually use this like a microscope. So you can zoom in and zoom out, and it's actually very useful. In this blood smear here, and this is still pretty low magnification, what I want you to see is that we have many, many, many red blood cells. All of these pink round-shaped structures are actually red blood cells. We've got several white blood cells, and you can see them right here. And then we've also got several platelets, and you can see the platelets there as well. So this is actually kind of nice. If we were in a class or in a lab where we had access to microscopes and slides, you would be looking at these yourself, but since we're not, we're actually going to be using some virtual histology. Okay. So revisiting that our need to knows, we saw that formed elements include our cellular elements, and we can divide the cellular elements into erythrocytes and leukocytes, the red blood cells and the white blood cells. We can divide the leukocytes into two large categories, granulocytes and agranulocytes. Within our granulocytes and our agranulocytes, we talked about the different specific leukocytes that are present there. We talked about the buffy coat, which is going to be the region in the centrifuge tube of blood where the white blood cells are found, and platelets, which are going to be the cell fragments. Hemoglobin, we said, was the oxygen transporting protein that's present in red blood cells. Polycythemia and anemia are two disorders of the blood that can occur and result in different numbers of red blood cells that are being formed. And hematocrit is actually a term that we use to describe the volume of blood that is devoted to red blood cells. The only term that we didn't actually get to was agglutination. This becomes important when we talk about blood typing. And agglutination, another way to think of this, is actually clumping. We talk about agglutination when we see this clumping that's seen when cells that possess certain antigens on their plasma membranes come into contact with an antibody that recognizes that particular antigen, and it causes them to sort of bind all together and clump. This is very much an immune system concept, but we do see it in blood typing as well. All right, so this brings us to the end of our short video. Go ahead and watch the second video about recognizing the formed elements, and then at the end of that, don't forget to take the lab quiz. Thanks very much.